Hello everyone, let us continue with the chapter Molecular Basis of Inheritance Part 17. Now we are going to learn about methodology of Human Genome Project. So there are two steps which is carried out. The first one you call that to be expressed sequence tags. Identifying all the genes that are expressed as RNAs, you call that to be express sequence tag, normally in short form ESTs. Okay, and the other method is sequence annotation. So in DNA, we have two sequences. One you will be learning about coding sequence, the other one you will be learning about non-coding sequence. So what are coding sequences here? One is coding sequence and the other one is non-coding sequence. So here, coding sequence is which codes for a protein. We have learned about the codons, right? Okay, but non-coding sequence, it does not code for a protein. So here in this method, express sequence tags, only coding sequence is involved. Okay, so here it is done with the help of coding sequence. And in sequence annotation, both coding and non-coding sequences are involved. Compare with these two methods, this sequence annotation is alone followed in this Human Genome Project. So what do they do in sequence annotation method? The whole set of the genome is being isolated from the cell, right? First it is separated and then the DNA is broken into many fragments and these fragments are inserted into the host. Why? To carry out the process. So here for sequence annotation we need a host. So what is the host used here? Bacteria and yeast. So bacteria is a prokaryote. We have learned. So which is called as a prokaryote? The one which has, what is that? So which is called as a prokaryote? If there is no well-defined nucleus, then you call that to be a prokaryote. So here bacteria is a prokaryote. Then what about yeast? Yeast is a eukaryote. So what is eukaryote? Well-defined nucleus is present in yeast. So these two organisms are involved in this process. They are used as a host. So what did they do? All these sequences, the whole set of genome is being isolated and it is broken into fragments and then these fragments are inserted into the host. How? It is used as a vector. So which is used as a vector? BAC. So what is BAC? Bacterial artificial chromosome. And YAC, yeast artificial chromosome. So these two chromosomes are used as vectors. So these fragments, they are inserted into the host, bacteria and yeast, with the help of these vectors, which is called as bacterial artificial chromosome, and YAC is called as yeast artificial chromosome. And one more methodology can also be done where you call it as PCR, polymerase chain reaction, which is used to make multiple copies of DNA fragments. Okay. Then what happened after inserting the fragments into the host, bacteria and yeast, with the help of these vectors, the DNA is being sequenced. It is annotated. So annotated DNA sequences are formed with the technique called as annotated DNA sequence technique. So what is the name of the technique? Annotated DNA sequence technique. So this technique was developed by Frederick Sanger. Who is he? Frederick Sanger. And he is a double Nobel laureate. Double Nobel laureate. So double, right? So one, he got Nobel Prize for this sequencing the DNA. For DNA sequencing, one Nobel Prize. The next one for studying the insulin structure. So he is called as a double Nobel laureate. So this annotated DNA sequence technique, it is developed by Frederick Sanger. So this technique was used here to make DNA sequences. And these annotated DNA sequences are used for computerized programs. Okay, so in that case, all the human chromosomes, so how many chromosomes are there? What did we study in last class? Haploid set of chromosomes, genome, N is 23. So 22 autosomes plus 1 sex chromosome. So if it is a female gamete, it is 22 plus X. If it is a male gamete, it is 22 plus Y. So all the human chromosomes have been sequenced. So how many chromosomes are being sequenced? Totally it is 24. So how come 24? 22 autosomes plus X and Y. So 22, 23, 24. So that is how total of 24 chromosomes are sequenced. Okay, and we have studied that. It is launched in the year, HGP is launched in the year 1990 and it was completed in 2003. So it is a 13-year project. Actually, the estimation was 2003. But here, the chromosome 1 was last to be sequenced in May 2006. Okay, so all the human chromosomes have been sequenced with the help of this HGP. So how many chromosomes are being sequenced? 22 autosomes plus X and Y, 2 sex chromosomes. So total of 24 chromosomes have been sequenced with the help of Human Genome Project. Next is salient features of human genome. So here the human genome contains 3164.7 million nucleotide bases. Nucleotide. We have learnt in DNA. What is a nucleotide? Which consists of sugar, nitrogenous bases, and then phosphate. So all together, nucleoside and phosphate joined together, you call that to be what? Nucleotide. Next one, the average gene consists of 3000 bases. The largest known human gene, what is the name? Dystrophin. The largest known human gene is dystrophin, okay, which consists of 2.4 million bases. Okay, and then almost if you take 99% of nucleotide bases are exactly the same in all people. If you take each and every individual, 99.9% of nucleotide bases are same. So only here small percentage it differs. That is why each and every individual look different. That is the reason this scientist have started sequencing the DNA. Right? And then what about the functions? Normally the functions are unknown for over 50% of the discovered genes. And then less than 2% of the genome codes for proteins. And next one, repetitive sequences. What do you mean by the term repetitive sequences? In satellite DNA, you have repetitive sequences. A sequence that are repeated 100 to 1000 times, you call that to be repetitive sequences. That is why I have mentioned here repeated many times. So they are called as what? Repetitive sequences. And these repeated sequences, when you take, it make up very large portion of the human genome. So most of the sequences will be repeated. Right? And then one more thing, this chromosome 1 has most genes. We have learned about 24 chromosomes. So 22 are autosomes plus X and Y sex chromosomes, 2 sex chromosomes. So including autosomes and sex chromosomes, you have 24. So out of 24, this chromosome 1 was found to be, what is the number of genes? 2968, which has most genes. And then what about chromosome Y, which has the fewest number of genes? It is 231. So chromosome 1 has most genes, 2968, and chromosome Y has fewest genes, 231. And one more thing you have to remember about SNPs. So what is SNP? Single nucleotide polymorphism, which is pronounced as SNPs, SNIPS, SNPs. So single nucleotide polymorphism. So what is the main function of this single nucleotide polymorphism? We can very easily diagnose the diseases. How to find out the human history? This single nucleotide polymorphism will be very helpful. So these are the salient features of human genome. Next is applications of Human Genome Project. So here first one, to determine the genes that change cancerous cell to the normal cells. So this is one possibility. Then to get designer drugs. So this Human Genome Project will also help you to get designer drugs and genetically modified diet, genetically modified food. So according to individual need. So that is possible. And one more thing, it has given huge data on various aspects of human genome. So 3 into 10 to the power 9 base pairs we have studied. So whole set of information, it is being sequenced. So along with this HGP, along with this Human Genome Project, so till now we have studied about human. But here, along with this Human Genome Project, a number of model organism genomes are also sequenced. So what are those organisms other than human? What are the genomes that are sequenced in which organism? If you see, example E. coli, Escherichia coli. Even in E. coli it is being carried out. And then Saccharomyces cerevisiae, it is yeast, fungus. And the next one is Senor habitus elegans. So what is this Senor habitus elegans? It is a free living nematode. Nematihelminthes. Helminth is a worm. Senorhabditis elegans. The next one is Drosophila melanogaster. It is a fruit fly. And the last one is Arabidopsis thaliana. Arabidopsis thaliana. It is a weed which has a short life cycle. So in E. coli, Saccharomyces cerevisiae, Senorhabditis elegans, Drosophila melanogaster, Arabidopsis thaliana. So in all these organisms also, the genomes are being sequenced. If you like this video, share, comment and subscribe my YouTube channel and follow me on Instagram and Facebook, and the link is available in the description box. Thank you.